Tonight, it's the 5th lecture already in our series, Frontiers in Genetic. I'm very pleased and honoured to introduce Dr. Aneil Agrawal, Distinguished Professor of Evolutionary Genetics in the Department of Ecology and Evolutionary Biology at the University of Toronto.
Thanks for the invitation. I think this is a wonderful series that you are running here. And I'm curious, how many people have been to at least one other lecture in this series? If you just put up your hands. Great, okay. I was hoping someone else would have done the prep work for me, so that sounds good. This is one of the youngest and smallest science departments on campus, and you probably don't know anything about it. So I want to take just a minute to tell you about the kinds of things that go on in my department, other than what something that I'll tell you about tonight. So this is Marty Krakozik. He's a population ecologist, and so he, among other things, is interested in quantifying the extent to which there's disease transmission from salmon farms to wild salmon populations, particularly the juvenile salmon as they leave rivers and they swim out to the open ocean past these salmon farms. They can become infected by these sea lice, and so he quantifies the extent to which disease is transmitted out of salmon farms into wild stocks, how that affects the health and fitness of juvenile salmon, and ultimately how it affects the population size and dynamics of the wild salmon stock that, is, I'm sure you all know, is an extremely important species to the ecology as well as to the economy and the culture of the West Coast, Canada. This is another of my colleagues, Nicole Medeo. She's a theoretical biologist, and she asks questions such as, why do some diseases evolve to be more virulent than other diseases? And she also works closely with experimental biologists studying malaria, who are interested in how the dynamics by which malaria multiplies within hosts. And so some of her theoretical models help guide experimenters in designing new kinds of experiments to learn about this disease. And this is Stephen Wright, who's probably my closest colleague amongst this group. He's an evolutionary geneticist who asked many of the kinds of questions that I'll refer to today with respect to humans, but he does it in plants. So understanding what are the forces that have shaped plant genomes, how much of the genome is under selection, and with some applications to things like herbicide resistance. That's occurring in crops and affecting agriculture. What do I do? I'm also an evolutionary geneticist. I don't work on humans. I ask questions such as this: why do organisms shuffle their genomes through sex and recombination? And that's the topic that I'll talk about tomorrow morning for any who can join for that. And I'm also, my other major interest is the one that Joanna mentioned, which is what is the effect of this constant influx of new deleterious mutations that are occurring all the time? How do they matter to evolution? But actually, Stephen and I, that's Stephen there again, Stephen and I co-teach a course on the evolution of the human genome, and I thought that would be of the broadest interest to people, so that's what I decided I'd talk about tonight. Okay, so what is a genome? I'm sure you've heard this several times before. It's very simply put, it's sort of the full complement of DNA that you have. And what is DNA? Well, DNA is these long, fancy macromolecules, and that's a bunch of biochemistry that I don't know much about, so I won't talk about that. The interesting bit for most geneticists is that you have this sugar phosphate backbone, but it's these connections between the two strands, the bases that join them, that sort of form the alphabet of the DNA. So we've got 4 bases: guanine, cytosine, thymine, and adenine, and they always go in pairs. G always goes with C, T always goes with A. And so if you know what's on one side, then you know what's on the other because they're always paired in that way. And so it's those 4 letters that are that forms the genetic code. Okay, and so DNA is you can think of it as this very long string of these letters, these G's, C's, A's, and T's, and some of that forms genes. So what are genes? Genes are these stretches of DNA that has a section that is essentially a tag that says, I'm the start of a gene. And a gene codes for a protein, and proteins are really sort of the action molecules of biology. And curiously, so this is a schematic that just represents a long stretch of DNA, and with this part representing a gene, a protein coding gene. And this part here contains the information to make that protein, given by the particular sequences of A, C's, G's, and T's. But curiously, it consists of 2 kinds of segments color-coded here, exons and introns. And what will happen is this promoter region is a region that says this is a gene. This gene gets what we call transcribed. A copy of it gets made. Instead of DNA, it gets transcribed into something we call RNA. And then these gray bits, which are these introns, get cut out. And the remaining part of it is what really codes the protein. That gets exported to a different part of the cell that then translates this RNA sequence into some functional protein. Okay? You don't really need to know any of that for most of what I'm going to talk about, but that's just a little bit of basic biology for you. So when we think about a stretch of DNA, and this might represent part of a gene, we could represent the DNA from different individuals for this particular gene by these sets of letters. And what you'll notice here is that everybody is the same. In this position, everyone has a G. In the next position, everyone has an A, has a C, and then an A, and then a C, etc. But there's what we call polymorphisms. Not everybody has exactly the same DNA. So here, this individual has a G where everyone else has an A, and at this position, these 2 individuals have C's and the others have T's. So one kind of genetic variant is when you just change one of these bases, or what we call a single nucleotide polymorphism. Or you might sometimes hear this word SNP, which is just the acronym for single nucleotide polymorphism. So that's one kind of genetic variant. And by the way, we use the word allele. You might have heard that word in some of the previous lecture, to just mean a different genetic form. So this individual has a G, we'd say it has a G allele, and this individual has an A allele with respect to this site. That's not the only kind of genetic variance we can have. Here's another stretch of DNA where we've lined up the DNA from all these individuals. And what you'll notice here is this individual is just completely missing a few base pairs. Or alternatively, you can view it as everybody else has some extra that one individual doesn't. Depends whether you're a half glass full or half glass empty kind of person. So it could be either an insertion or a deletion. So we sometimes call those as indels. I'm just showing you sort of 2 kinds of forms that genetic variants can take. They can be just a different base pair, or you can be missing a piece of DNA that others have. Okay, so as an evolutionary biologist or a population geneticist, we're interested in the frequency of different genetic variants in a population, how they change over time. Whoops, so let me just go back. So, again, in this example, we have one copy of the G at this site at this point in time. But maybe this G allele confers a large advantage, and if you were to sample the population later in time, we would expect this G allele to increase in frequency. So instead of it being 1 out of however many copies there are here, 8 or 10 or something, in some future time it might be 5 out of 10 or 10 out of 10. And of course, the term that's almost synonymous with evolution is natural selection, but they're not synonyms. Evolution from a population geneticist point of view is anything that causes allele frequencies to change over time. The most famous evolutionary force, the one that Darwin brought to the world formally, is natural selection. And so we could imagine if it's some rare mutation that happens. When it first happens, it exists as a single copy out of the entire population, and if it's favored, it will start at a frequency close to 0, and it will increase over time until it's at a frequency of 1. That is, everybody has that variant. But that's not the only evolutionary force, and the other one that I want to talk about today, in addition to natural selection, is genetic drift. And genetic drift is basically chance. But it's a kind of, well, we'll see. So here, I'm going to represent alternative variants just by big A's and little a's. They could be any kind of genetic variants, but now we are diploid organisms, which means we have 2 copies of pretty much every gene. Here we have a little population, a population of 4 individuals, 1, 2, 3, 4. Each one has 2 copies of the gene. Most individuals have the little a version of the gene. So little a, little a, little a, little a. But this one individual has little a and a big A. And they reproduce, so these 2 individuals mate and produce 2 offspring. Now, these individuals only have little a's, so they only transmit little a's to their offspring. These 2 individuals mate and reproduce, and by chance, this individual gives the big A copy to both of its offspring. Right? Just by chance. There's a 50-50 chance of, for every gene you have, or most every gene you have 2 copies. A copy you got from your mom, and a copy you got from your dad. And if you have offspring of your own, you transmitted your mom's copy or your dad's copy with equal chance. And so that means if you have had 2 kids, you could have given them both of them your mom's copy or both of them your dad's copy. So just by chance, you can transmit the same copy twice. So from a population geneticist point of view, with this very artificial population, when I looked at it at time 0, this first line here, there was 1 out of the 8 copies of this gene. 1 out of the 8 copies was big A. So we'd say the frequency was 12.5%, 1/8. But just by chance, it increased to 25%. And we could go one more generation, and this time, this guy apparently gets around because he mated both with this individual and with this individual, producing a whole bunch of offspring. This guy may have mated but didn't leave any offspring. And so this allele managed to now increase to 50%. Not because it confers any intrinsic advantage, just by chance. So we've seen this allele, just by chance, is increasing. Now the next generation, that allele, by chance, doesn't do so well. And it goes back down in frequency. The allele frequency can bounce up and down. And so we can get changes, and it can go from 0% to 100%. Now, these changes, what we call genetic drift, are more extreme in smaller populations. And this is why this kind of stuff becomes a concern with respect to conservation biology. But this is an extremely important force in understanding genomes and how they change over time. Natural selection is the force that we think about when we think about evolution, and it's certainly the force that drives adaptive evolution, but not all evolution is adaptive. And especially in the genome, as we'll see in a few slides, a lot of changes aren't adaptive. They're not good, they're not bad, they're nothing. But the genome can change by chance. I should also say that in this example, I've said this alternate version of the gene confers no selective advantage or disadvantage. It's what we call selectively neutral. And genetic drift is the primary determinant of the fate of neutral genetic variants. But genetic drift also affects variants that are under selection. So this was the picture I showed you before, where natural selection just causes this beautiful, smooth increase in the frequency of the gene across a number of generations. But that's the way it would work in an infinite population. So for those of you, I know there's some engineers and such in the audience. So this is how things would work in an infinite population. But in a finite population, there's genetic sampling that occurs every generation that causes that genetic drift. And so here, this represents a stochastic simulation where you have a mutant that has a slight advantage. And you start it at 50% in this simulation, and you do different runs of this and say what will happen to it over time. It confers an advantage, so most of the time it increases in frequency, but often it's bouncing up and down. We started at 50%, so oftentimes it goes below where it initially started. Sometimes it just gets completely lost. And what this means is that in a finite population, beneficial mutations sometimes get lost. Remember, in a natural case, a mutation wouldn't start at 50%. Not half of the individuals would start with a new mutation. It occurs once. It's in one copy. And so a lot of the time, things get lost just by chance. And what that also means is bad mutations, sometimes despite selection acting on them, can spread all the way to fixation. Okay? And there's a robust mathematical framework for understanding this process. Okay, so that was some sort of basic evolutionary principles. Now I want to get to talking about genomes. So what's in the human genome? So here's a sort of cartoon of the human genome. So our genome is made up of 23 chromosomes. 22 of those are what we call autosomes. And we have 2 copies of each autosome, a copy we got from mom and a copy we got from dad. And then one of those is a sex chromosome. And so, of course, females are XX and males are XY. And there's some really interesting evolutionary biology about why this happens. But I don't have time to talk about that today. Okay, so the human genome was first draft sequence of it came out in 2001, so 15 years ago now. But that was really sort of sort of viewed as sort of the first piece of a much larger puzzle. First of all, the draft was a draft. We didn't have a complete sequence, and even now it's still hard to fully sequence the genome. Some parts are very hard to assemble. And we don't really understand, there was still a long way to go to understand what the sequence meant, and we're still unraveling that. So what I want to talk about today is why is our genome the way it is, and what can we learn from it? And so I want to consider the human genome in an evolutionary context at 3 levels today, in sort of three somewhat separate parts of the talk. So the first is thinking about our genome on the grand evolutionary scale. How does our genome compare to genomes of all other organisms? Second, I want to talk about how does comparing our genome to our closest non-human relatives and what we can learn from doing that kind of a comparison. And finally, I want to talk about what we can learn from looking at variation in genomes amongst humans. Okay, so if we're going to think on the large evolutionary scale, I'm going to remind you that our genome is a product of biological evolution that's been occurring on this planet for about 3.5 billion years. And so this is the sort of the tree of life at the largest scale. We have the archaea, the bacteria, and then these are most of the kinds of organisms that we think about on a daily basis: plants, animals, and fungi. And we can zoom in on that to the animals, and there's hundreds of millions of years of evolution that reflects all of the animals, and we're embedded in just one little branch of that evolution. These are the chordates, and the majority of the chordates are the vertebrates, the things with backbones, and they're the mammals that split off from the reptiles some 300 million years ago, and that it was really when the dinosaurs went out of the picture after that mammals really diversified into all of the groups that we see today. That happened some 65 million years ago. And one of those branches is the primates. And primates have evolved, and we split off from chimps. That's thought to be about 6 million years ago. And it's worth pointing out that our closest relatives are chimps. And chimps are more closely related to us than they are to orangutans. Okay? So a lot of people don't know that, but okay, so let's think about a genome. If you were an engineer and you were building a genome, you'd say, I need some genes, and they have to code for these proteins, and I might need some regulatory material around those genes to tell those genes when they should get expressed and so on. But that would be it, more or less, in a nutshell. And you might have these really these genes that were really simple. They're just gene after gene after gene with space, with a little bit of space between them that contained a bit of regulatory material. That might be how you would imagine a genome would look. And you might say, well, to build a more complex organism, I might need more genes or more regulatory material, so the genomes might be bigger for more complex organisms, and that's the way things should fall out. But people started figuring out about 50 years ago that there was more to the story than that. So this is actually an amazing plot made by a colleague at Guelph. This shows genome sizes in terms of millions of base pairs on a log scale. So one means, or 0 means a million, and 3 means a billion. And so this is showing the ranges of genome sizes within these different groups. So let's see, so here's the fungi, and look at this. So they go from 10 million to a billion in base pairs, their genomes. So there's a huge amount of variation amongst fungi in how big their genomes are. Here's another group here, the annelids, so these are the worms. Look how big their genomes are, and how much variation there is, right? Orders of magnitude. Across this whole figure, there's about 6 orders of magnitude in how large those genomes are. Massive variation. And here's mammals up at the top. And mammals, that's, of course, our group. We have big genomes, but we certainly don't have the biggest genomes. And in fact, the star represents us. We're sort of smack dab in the middle of mammals. We don't have particularly large genomes or particularly small genomes with respect to other mammals. And we certainly don't have big genomes compared to salamanders or lungfishes. So, look, that's how big our genome would be relative to a lungfish genome, just to give you a sense of scale. So If you're feeling pretty important right now, then this should cut you back down to size. Okay, so so why is that? Why do these genomes, why do they vary so much in size? And it doesn't seem intuitively obvious that it has to do with complexity of the organism, right? Probably most of you don't believe that you are this much less complex than a lungfish. So that was a mystery, and it still sort of is somewhat of a large mystery of why there is so much variation. But now we understand. In the next slide, you'll see we're starting to understand what's in these genomes. Is it just genes? If we were just designing genomes, it would be just genes. But it turns out that's not the case at all. Okay, so here's a pie chart showing what the genome is made of. And there's a lot of stuff on here that you won't know what it is. But here's the one part that you do know. Genes. Genes make up about 1.5% of our genomes. Most of your genome is not genes. And in fact, a large fraction of it, 45% of it, is what we call transposable elements or mobile elements. And I'll return to that in a minute and tell you more about that. But I want to show you this table that highlights some numbers from very well-studied organisms in biology. So this is yeast. This is baker's yeast that's extremely well-studied. This is Arabidopsis thaliana. It's a little weed from the mustard family that is kind of the fruit fly of the plant world in the sense of it's very intensively studied. This is C. elegans, a little roundworm, another sort of model organism in genetics. Drosophila melanogaster, that's the fruit fly that's been famous for many years in genetics. Down here is mouse, another model organism for medical and research and genetics, and Homo sapiens is us. And so across these model organisms of biology, what I call our favorite model organisms, genome size goes from, in yeast, being 12 megabases up to us, which is 29 megabases, or about 3 billion base pairs. So that's about a 200-fold variation in genome size. So that's genome size. What if we look at gene number? Well, gene number in that smallest genome is about 6,000 genes. In our genome, it's about 24,000. In other words, a 4-fold variation. So there's 200-fold variation in how large the genome is, but there's only 4-fold variation in how many genes there are. Right? So most of that variation that I showed you in that earlier slide across all of the diversity of life, there's massive variation. Most of that variation is because of the stuff other than genes. Right? Remember, genes only make up a small fraction of our genomes. In some other organisms, it makes up a large fraction of their genome. But what's really varying is how much other stuff there is in genomes. And that's why that's why you shouldn't feel inferior to a lungfish. It's not that you have so many fewer genes. It's that their genomes are filled with other stuff. Okay, so what is that other stuff? 45% of it in our genome are what is known as these mobile element insertions or transposable elements. And there are two in particular that are very common that I'll just tell you about briefly because I think their biology is really interesting. This L1 and this ALU elements. So what are these things? So these L1 elements are really cool. So a functional version of it contains 2 genes, so that's what these colored segments represent, these 2 genes. And it has a promoter region, which means that this gene gets a copy made of it, Just like I showed you with earlier, a gene has a promoter region that says, I'm a gene, make a copy of me so I can get translated into a protein. And so that's what happens. It has copies get made of it, and it contains a special gene. And that special gene is what we call a reverse transcriptase. And it takes that messenger RNA, so this gets transcribed into this messenger RNA, and will get made into a protein. What the protein does is it finds other copies of it in that messenger format and retakes it back to some new place in the genome and makes another copy of itself. It doesn't do you any good, but it increases its own abundance. Okay? So natural selection can play out at many levels. And it's being played out in our genomes to no advantage to us and often to considerable disadvantage, because it might, when it reinserts, it might reinsert in the middle of a gene you actually need. In which case, you're out of luck. These are sometimes called jumping genes, and they were famously discovered by Barbara McClintock in maize and corn. So we have in our genome about 500,000 copies of this element, which an element is about 6,000 base pairs long. We have about 5,000 copies of it because it can copy itself. Now it's bad at copying itself, so almost always when it copies itself back into the genome, it makes a mistake and it's no longer functional. So almost all of these 500,000 copies are what we call dead elements. They're not capable of recopying themselves. But there are about around less than 100 functional copies that are still active. These elements are sort of genomic parasites. But this is one of the two elements. The other elements is interesting because it's actually a parasite of the parasite. It's missing the gene that copies itself back in. But what it does is it uses this parasite's genomic parasite, makes the gene that copies it back into the genome. It sort of hijacks it and tricks it into copying it into the genome. And we have about 1 million copies of that one in our genomes. But only a few of them are active. Okay, so that's what most of our genomes are made of. Okay, so next I want to talk about what we can learn from human-chimp comparative genomics, so comparing our genomes to those of chimps. So first, a couple basic facts. So if you compare our genomes to those of chimps, you'll remember that early on I showed you lining up the DNA, the A's and the T's across a set of individuals. Well, imagine you do that for a human and for a chimp. When you line them up, it turns out they're going to be the same at 99% of sites. About 1 out of every 100 sites is going to be different, with a caveat. And the caveat is where you can actually line them up. There's some regions where we have DNA that they don't, and vice versa. And so there's about 3%. There's about a 3% difference in what DNA we have. So you'll sometimes hear this in the news that we're 99% the same as chimps, and that comes from the part where you can actually align it, and but there's actually a much, compared to that, a 3 times larger difference in what parts can be aligned. But even if you add that all together, still we're very similar. Okay, so it turns out by comparing our genomes to those of chimps and using some evolutionary thinking, we can ask some really interesting questions. And this is the one I want to talk about. How much of our genome is under selection? So I showed you we have this huge, we've got this genome. Only 1.5% of it is genes. And people figured that out some time ago, but they didn't know what's going on with the rest of that genome. Is it important? I mean, maybe all of that other stuff that's not protein coding genes is somehow involved in regulating the expression of those protein coding genes. So how much of our genome is under selection? Is it just the 1.5% that actually makes up the genes? Maybe it's less than that, because maybe a lot of those genes, you can change them a bunch and it doesn't really matter. Maybe only certain parts of those genes are really, really important. So maybe it's less than that 1.5%. So how constrained are the gene parts of the genome? But maybe it's more than that. Maybe the non-genic parts are really important. And of course, we're humans, right? We're not fruit flies, so in fruit flies, we can do experiments where we manipulate parts of the DNA and ask, what's the effect? You're not allowed to do that, humans, for good reasons, right? So how can we figure this kind of thing out? Well, we can gain insights into this using a comparative evolutionary genomics perspective. So here's the idea. A sort of cartoon version of this idea. So imagine we have some neutral sites, so sites that are not under selection. And you'll just have to trust me that we have a way of identifying a set of sites in the genome that we're pretty sure are not under selection. So if we take if we look at 1,000 of those sites for humans and compare them to the comparable sites in chimps. And so when you look at those 1,000 sites, these colored bars represent mutations that might have occurred at those different sites. And so 4 mutations happen to occur in the human lineage and another 4 in the chimp lineage. And when we compare the human to the chimp, we would find 8 differences out of the 1,000 when we look at those 1,000 sites. And that reflects the neutral mutation, that reflects just the mutation rate. So now we can do the same thing for sites that we know that if you change them, it would affect a protein, the protein produced by a gene. And so we could look at 1,000 protein affecting sites, and the mutation rate should be the same. So we would again expect to see 8 mutations to have occurred over that time period for those 1,000 sites. But if selection is acting, most changes are going to be bad, right? I mean, so if you think about, most organisms are sort of pretty well-functioning machines, right? So if you change something at random, it's bad. When I tell my students this, I say, here's a little experiment if you don't believe me. Tonight when you go home, open up your computer and change something at random and see if it worked better or worse. I'm not responsible for what happens because I've told you what I think will happen, right? But most changes are bad. So what that means is we expect those 8 mutations to have occurred, but selection would have prevented those from persisting, except for the changes that don't really matter. Okay? So ignoring any adaptive evolution, which for this kind of analysis, to a good approximation, those adaptive changes occur rarely enough. We'd say only two persist. That means 2 out of the 8 that actually occurred, only 2 were neutral. The other 6 out of the 8 were selectively constrained sites. Mutations occurred there, but they couldn't persist through evolution. So we would say that the protein affecting sites evolved 25% as fast, 2 out of 1,000 rather than 8 out of 1,000. They evolved 25% as fast as the neutral sites. We say there's a constraint of 75%. And so this is a cartoon example, but that's about what the number is. It's about 75% of constraint in the genes. Okay, but remember, most of the genome is not the genes. So what happens elsewhere in the genome? So you can do the same kind of thing, and this is a graph of a measure of constraint as a function of distance from a gene. So this is looking what we call upstream of a gene. So the gene starts at 0, and this is looking at base pairs upstream of the gene, and from many, many genes, and this is downstream from genes, data from many genes. And what you see is there's some higher level of constraint closer to the genes, approaching 10%. But as you walk away from the genes, so 10,000 base pairs away or 20,000 base pairs away, constraint goes down. So there's some level of constraint that probably reflects that the regulatory machinery for genes is close to genes, and as you walk away, there's less constraint on the genome. So what does this mean overall? This means that probably about 5% of the non-coding, non-protein coding parts of the genome are under constraint, and almost all of the genome is non-protein coding. So what that means is sort of over the genome overall, it's probably about 5 to 8% of the genome are our current estimates of how much we think sort of matters with respect to natural selection. The other 90 to 95% of the genome doesn't matter. And this is an interesting result in contrast to other work. So molecular biologists who don't think at all about evolutionary work, not all molecular biologists are like this, but there's a large set of them, they recently came out with an estimate of 80% of the genome being functional. And it was actually all over the news a year ago. Actually, it came out in the news about 3 days before I gave this lecture. And so I thought, uh-oh. And then I realized, no, they're crazy. And because of, what's their measure of functional? It's essentially, is there any evidence that that part of the genome is transcribed or interacts with something? I was telling this story to my wife the other day, and my wife's a chemist. She's like, they're molecules. Molecules interact. It doesn't mean that it's functional or important. And that was her reaction as a chemist, and my reaction as an evolutionary biologist is, those numbers don't make any sense because we would see evidence of evolutionary constraint occurring much more broadly in the genome. The molecular biologists have since somewhat walked back that number. Okay, so it seems like much of our genome is probably not selectively constrained, not functionally important. Okay. So the last section, so in that section I was telling you about things we can do comparing genomes from different species, humans and chimps. But we can also think about variation within humans. And this is the field of population genetics, or now we put omics on everything, so it's population genomics. And this is population genomics is the study of within species sequence, DNA sequence variation, both within and among local populations of humans. To introduce this to you, I want to use this little schematic. So this represents a little stretch of DNA from a bunch of individuals. So it's just like when I had the A's, the T's, and it's just the same as that, although I've simplified the figure because I'm highlighting the spots where there are differences amongst the individuals. Where it's black, everybody has exactly the same sequence. Okay? So what we see is this one individual has a different variant at this site. These 3 individuals have a different variant at that site, and so on and so forth. So imagine all of these variants were neutral. They're selectively neutral. They confer no advantage or disadvantage. But one day, whoops, I'm pushing the wrong button again. One day, a mutation occurs, this red point here, that's beneficial. And this beneficial allele confers an advantage, and it starts increasing in frequency in the population. And so what might happen, oh, right, so sorry, let me before we introduce that. The point here is that there's a bunch of there's a bunch of neutral variation amongst this sample of individuals. And the amount of that variation reflects the mutation rate and genetic drift. But then we get this beneficial mutation, and it will increase in frequency. And when it increases, so selection is favoring that red point there. But look what happens. By making copies of this, it's actually making copies of all of that. And so at some time in the future, what you might see is no variation. Right? Because this thing sweeps through we call it a selective sweep. It increases in frequency, sweeping out the variation. So you see colored points here, but those colored points no longer represent variation because everybody's got them. Right? It's not variation because everyone's got the same thing. Not everyone had the same thing here. Right? Eventually, new mutations will occur and reintroduce variation. But when a selective sweep occurs, it wipes out variation, sort of. Okay, so I'm going to come back to that sort of right now. So I need to tell you something else about basic genetics in case you don't already know. So that's recombination. So imagine this is one of my chromosomes, and I have some genetic, and this is the copy of my chromosome I got from my dad. And I have some interesting variant there. Or maybe it's not interesting, it's just a variant, okay? And my dad had some other interesting variants that he gave me, and they're here and here. So it turns out that I pass on this variant to my son. But actually, remember, I don't just have one chromosome. I don't just have one copy of this chromosome, the one I got from my dad. I also have the one I got from my mom. And when I produce offspring, there's this phenomenon known as recombination that causes, essentially, you swap parts of chromosomes, equivalent parts of chromosomes. And so what I actually give to my son is not a whole version of my dad's chromosome, but I gave him that part of my dad and then this part from my mom. Okay, so remember, there was this other interesting genetic variant from my dad, but that's not what my son gets. He gets this one and this one, but he doesn't get that one because of recombination. And my son, of course, gets a chromosome from my wife, from his mom. That's the one he got from his mom. And when if he has a kid, and if he were to transmit that, there might be a recombination event that separates that out. The key point that I want you to get from this is any genetic variant at one site starts on a particular, what we call a particular genetic background, but gets separated from that background over time across generations because recombination is mixing chromosomes from different parents. Okay? So now let's go back. Now I want to go from thinking about one individual in a family, to thinking at the sort of population level and thinking about those selective sweeps that wipe out diversity. So how does that translate up? Okay, so this is what we saw before. We get this favored genetic variant, and it increases in frequency, wiping out all the diversity around it. Well, that's what would happen if there was no recombination, but there is recombination. And so what happens is you do lose diversity close to the selected site. So the selected site, again, is shown in red. And so we've lost the diversity that was around that. Right? Everyone's got this black, because that's what this black point was with it initially. And we lost them. But there's been some recombination events. So we've managed to save a variant there and a variant there. And as you sort of go further away from the target of selection, you're going to maintain more of the variation. You will not see this massive reduction in variation. Okay, so the point of this is that these are signatures of selection. when we look at a chromosome, at a region of a chromosome across a bunch of individuals, and we see all of a sudden in one region there's very low levels of diversity, that's a signature of selection in the past, in the not too distant past. That's sort of the simplest signature. There's a variety of signatures we can look for. There are some that are more subtle that I won't go into in detail. The point is that we can look at patterns of genetic variation to identify targets of selection in the not too distant past. So I want to tell you about one or two studies that have done this with human populations. So this is data from a study where they looked at some genome sequences from three groups of people. They had some genomes from Europeans that are going to be shown, I think, in these purple bars, and from a set of Chinese individuals in blue, and then in red, which is where the interesting stuff comes from, is from a group of native northeastern Siberians. And so they looked across the genome. So this is a representation of chromosome 11 in humans, and a zoom in on one particular part of chromosome 11 where they found some interesting signatures of selection. And you'll note that these are regions where you only where you see high bars or large bars is where you see a pattern that doesn't seem like it could have arisen just by chance. It's too extreme to have arisen just by chance. And so those are areas of interest for us to look at. And this is one where this is a place where you only see it in the northern Siberians, the red bars. You do not see it in the Europeans or the East Asians. Along here is sort of that maps onto these positions are where genes are located. And where this region is a gene called CPT1A. Okay, so from looking at these signatures, there's evidence of strong selection, and I won't go into how, but they were able to make an inference that selection was strong, and this mutation probably occurred in the last 6,000 to 20,000 years. So what is this gene? What does it do? So we can rely on our molecular biology colleagues who really study functionally what genes do, and they tell us that CPT1A is involved in it's a key regulator of mitochondrial long-chain fatty acid oxidation. In other words, it is involved in fat metabolism. And this derived allele, the allele that exists that's common in this northeastern Siberian population was already known to have some health effects. So it's associated with hypoglycemia and high infant mortality. As I said, yet despite the fact that it has these negative health effects, it's found at high frequency in these northeastern Siberians. And it shows the signature of strong positive selection on it. It was favored. What our inference is, before people went and asked what does it do, was that it was favored by selection. Well, the hypothesis is that actually this alternative allele that causes negative health effects, particularly with modern diets, was probably strongly beneficial in the high-fat, low-sugar diet that was traditional for the people there. Okay? And now here's a similar story that might affect you more directly. So here's another study that this study focused primarily on Inuit people from Greenland. And they, again, used sort of similar kinds of approaches to identify genes that had been under strong positive selection. That is, they were favored by natural selection. And one of the genes they found that was most strongly favored are these FADS genes that are these fatty acid desaturases that are involved in processing of omega-3 and omega-6 fatty acids. I'm pretty sure almost everyone in this room has heard of omega-3, right? So when this paper came out, so this thing's favored by selection, but it's only in these Inuit people at a high frequency, this variant of the gene. Okay, that turned out to be quite interesting. It was written up in the New York Times by a popular article on this scientific result by Carl Zimmer, and I just want to read this little quote. In the 1970s, Danish researchers studying Inuit metabolism proposed that omega-3 fatty acids found in fish were protective against risk of heart disease from eating high-fat, high-protein diets. Those conclusions eventually led to the recommendation that Westerners eat more fish to help prevent heart disease and sent tens of millions scrambling for fish oil pills. I suspect there's a few fish oil pill consumers in this audience.
Krill.
But the problem is, you don't have the Inuit genetic variant of that gene. So what we suspect now is that adaptation by the northern peoples to this fat-rich, low-sugar diets has led to selection for more efficient processing of these omega-3s and omega-6 fatty acids. And so it's becoming clear that we need to incorporate the history, the adaptation history when making dietary and medical recommendations. In other words, omega-3s might not be so good for everybody because not everybody has the right genes to deal with it. And so this is sort of a bit of another entree into what you may have heard of before as personalized medicine, that the medical recommendations for people might be unique to your ethnic background, or even narrower than that, and unique to your particular genotype. So I want to end by briefly mentioning, we're doing there's a lot of work being done on understanding the genetic basis to complex diseases. So that means diseases that aren't caused by just a single big mutation. And really there's sort of two categories that things could fall into. One is known as the common disease, common variant hypothesis. That is where there's a set of alleles that are common, reasonably common in the population that play a role in causing the disease. And we're pretty good at finding those because they're common enough that we can associate a particular genotype with a disease, that we can link them that way. But the question is, why are they so common? And it may be that the reason they're common is because they were beneficial in the past, at least in some populations, in some contexts. But the other genetic cause of disease is not from the genes like the ones I was just showing you from the Inuit people, where it might be very beneficial for them to have those, but rather there is a constant influx of new mutations that are occurring all the time, and selection eliminates them, but it doesn't eliminate them instantly. Otherwise, those would be known as lethal mutations, right? So a mutation that increases your risk of heart disease by 1%, will take hundreds of generations to eliminate on average. In fact, we all carry many deleterious mutations that are not good in particular contexts. They're bad, and they're bad all the time, but they're just slightly bad, and so they persist for a while. So I will end just by, again, saying what have we learned today? Well, we've learned comparing our genomes to all other organisms that we've got big genomes, but we certainly have far from the biggest genomes. That we have a lot of genes, but we don't even have the most genes. There's other things that have more genes than us, but we're close to the top of the list, at least on that. And we also learned that most of our genome is not made of genes, and that these parasitic elements, these mobile elements, make up a large fraction. From comparing our genomes to chimps, they're really very similar. We can use that comparison to make inferences about how much selection is acting across the genome, and probably less than 8% of our genomes under selection. And we can also study variation within humans to get insights into human history. I didn't talk about that today very much, but and also about adaptation to different environments and what that means for health today. And with that, I will end by thanking you all for listening and invite you to come tomorrow morning if you're able. Thank you very much.